why is breathing so essential to life? What happens when we breathe? Okay, wait. The main thing starts from here only. All the energy required for life processes. Life processes are all those processes that are required to maintain maintain us living, such as breathing, eating, circulation, excretion. All these are the life processes. So all the energy required for life processes is obtained by the oxidation of some macromolecules that we call food. Oxidation here means breakdown. Now, only green plants and cyanobacteria can prepare their own food because they are autotrophs, and they do so with the photosynthesis process. They trap light energy and convert it into chemical energy. Now, we have learned in the previous chapter that these organisms that can perform photosynthesis, they can do so because of the presence of chlorophyll in them, right? Now, when those macromolecules or just in general biomolecules are broken down, a lot of energy is released because, breakdown of biomolecules in general, it doesn't matter which biomolecule is being broken down. When it in general is broken down, a lot of energy is released. And what actually do we break down? We break down the chemical bonds that are present between the molecules, such as, for example, carbohydrates such as the glucose, sucrose, and starch. They have carbon bonds in them. And when those carbon bonds are broken, a lot of energy is released. Is it clear?
Yes.
Now, in the plants, all of the cells photosynthesize because we know that the plant's body contains chlorophyll. Now, they carry out the photosynthesis. Hence, in green plants, all other organs, tissues, and cells that are non-green need food for oxidation. Now, for all those green parts, we know that, okay, they can make their own food. But what about the non-green parts? So they would require the food or the energy for the oxidation process. So food has to be translocated. Translocation means movement of food from where it is synthesized to where it is needed. That is translocation. Now, animals are heterotrophic, meaning they cannot make their own food and they are dependent on the plants for their food requirements. So they obtain food from plants directly if they are herbivores, or indirectly if they are carnivores, because carnivores eat the organisms that eat the plants. Saprophytes such as fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is inspired for life processes comes from photosynthesis. Now, in this chapter, we're going to deal with the cellular respiration, meaning the respiration at the cells level is cellular respiration. It is the mechanism of breakdown of food materials within cell to release energy, and the trapping of this energy for synthesis of ATP. Because ultimately, whenever the body requires any form of energy, that comes from ATP. Okay, so that is why everything will be coupled to ATP synthesis. Now, photosynthesis, of course, takes place within the chloroplast in the eukaryotes, whereas the breakdown of complex molecules to yield energy takes place in the cytoplasm and in the mitochondria, also only in eukaryotes. Because see, first of all, prokaryotes do not have these organelles only. Secondly, in the eukaryotic organism, since these are the organisms, for example, mitochondria, it is the chief, you can say it is the powerhouse of the cell, right? And how is it so? Because ATP synthesis takes place. Secondly, all the aerobic respiration takes place in mitochondria itself. Now, the breaking of carbon-carbon bonds of complex compounds through oxidation within the cell, leading to release of considerable amount of energy, is known as respiration. Now, the compounds that are broken down or the compounds that are oxidized during the process, these are the respiratory substrates. So, for example, if we are breaking down glucose molecule in order to release energy, then glucose becomes the respiratory substrate. Now, usually carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substances in some plants under certain conditions. The most preferred source for energy is carbohydrates. Always remember that. The most preferred source. But apart from that, there are other biomolecules that can be used as well. Now, during oxidation within a cell, all the energy contained in the respiratory substrate is not released free into the cell or in a single step. It is released in a series of slow stepwise reaction. It does not happen all at once. It happens in steps so that energy is collected at every step, and then when we combine it, it becomes a huge amount of energy. So that is why it happens in multiple steps. And then all of the energy which is released by the breakdown, it is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be used directly, but it is used to synthesize ATP. So wherever energy is released, it will be used to make ATP first, and then only the energy can be utilized. Now, whenever ATP is broken down, whenever and wherever energy needs to be utilized. And when ATP breaks down, what do we get? After the breakdown of ATP, what is the thing that we get? Like two components.
ATP and NADP.
No, no, ADP and inorganic phosphate, because
Inorganic.
yes, because ATP is made up of ADP plus PI. PI means inorganic phosphate. This is what we will get when ATP is broken down, and reverse of it will be for ATP synthesis. Now, hence, ATP acts as the energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring processes of the organisms, and the carbon skeleton produced during respiration is used as precursor for biosynthesis of other molecules in the cell. Meaning, this breakdown, this respiration is actually helping in two processes. The first one is to synthesize ATP. The second is to actually biosynthesize for the production of other molecules with the help of that substrate. Up until now, any doubts? This is the introductory part. Do you have any doubts up until now?
No, ma'am.
Everything is clear?
Yes.
Wherever you do have doubts, you can stop me, okay? Now, do plants breathe? Yes, plants require oxygen for respiration to occur, and they give out carbon dioxide. Hence, plants have systems in place that ensure the availability of O2. Plants, unlike animals, have no specialized organs for gaseous exchange, but they have stomata and lenticels for this very purpose. There are several reasons why plants can get along without respiratory organs. First of all, each plant part takes care of its own gas exchange needs. This is very little transport of gas. There is very little transport of gases from one plant part to another. The second reason, plants do not present great demands for gas exchange. So that is why they do not have the respiratory organs only, just like how we have lungs and nostrils and everything, they do not have. Roots, stems, and leaves respire at rates far lower than animals do. Only during photosynthesis are large volume of gases exchanged, and each leaf is well adapted to take care of its own needs during these periods, because the leaves, they have stomata present in them, and stomata is like sufficient for the plant's body. When cells photosynthesize, availability of oxygen is not a problem in these cells, since O2 is released within the cell only, because oxygen is a byproduct of photosynthesis. Now, the distances that the gases must diffuse even in large bulky plants is not that great. I mean, it's not huge distances. Like in our body, the gases have to be traveled a lot. We have so many cells, we have so many organs in our body. So the oxygen has to reach each and every single organ. But that's not the case for plants. Now, each living cell in a plant is located quite close to the surface of the plant. This is true for leaves, you may ask, but what about thick woody stems and roots? The living cells are organized in thin layers inside and beneath the bark. They also have special openings called lenticels. You might, if you have seen the, the pictures of bark of a tree, you might have seen something like this, usually in
Yes,
pictures.
ma'am.
Right?
Yeah.
Now, if you look very closely, you'll find tiny holes present on the bark. Those are lenticels. Now, lenticels are the special openings that are there for gaseous exchange. They're very important and they help the bark of the stem to actually exchange gases. Clear?
Ma'am, do stem have tomato for the gaseous exchange?
Hmm. The stem, if you're talking about the thin, flimsy stem, the normal plant stem, they have stomata for gaseous exchange. But we're talking about the stem, like the bark of a tree, which is very thick, right? And it's just so hard to cut.
Yes.
Yeah, so for that, stomata is present like in the green areas, isn't it? Because it is there in the chloroplast, it is there in the photosynthetic apparatus. But as far as the bark is concerned, see the trunk of the tree, it is mostly made up of dead tissues only because it has to be very supportive in nature. So that is why they have lenticels for extra support for gaseous exchange. Clear?
Yes.
Okay. The cells in the interior are dead and provide only mechanical support. Thus, most cells of plants have at least a pair of their surface in contact with the a part of the surface in contact with the air. This is also facilitated by loose packing of parenchyma cells in the leaves, stems, and roots, which provide interconnected network of air spaces. Because parenchyma, we know that intercellular spaces are present in them, isn't it? Do you remember from anatomy?
Yes, ma'am. It's present.
Very good. So that is why the parenchyma, which is present in the leaves, the stem, and the roots, they would have the proper connection of air spaces. Now, the complete combustion of glucose, which produces CO2 and H2O as end products, yields energy, most of which is given out as heat. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step, but in several small steps, enabling some steps to be just large enough that the energy release can be coupled to ATP synthesis. This is the reason why all the processes that takes place for the respiration, they are not done in just one large step, because if it was done in one large step, all the energy will be released in the form of heat. So as to not let that happen, it happens in a series of smaller steps, so that each one of those steps can be like, one step happened, the energy is saved for ATP synthesis, again another step happened, the energy is saved for ATP synthesis, and so on. With that being said, then comes a lot of a greater energy which couples to ATP synthesis. Clear? Also, do you know the meaning of catabolism? Catabolism and anabolism, two things are there. Do you know the difference between them?
Catabolism is a process where energy is released.
Where? I can't hear you, dear. Can you speak again, please? Catabolism is a process. See, have you heard about metabolism?
Yes, ma'am.
Hmm. This metabolism can actually be divided into two parts, that is catabolism and anabolism. See, catabolism is the process wherein the breakdown of stuff happens and it is accompanied by the release of a large amount of energy. So any molecule, if it gets broken down, then that is catabolism. But if any molecule is synthesized, for example, synthesis of ATP, so during the synthesis of any molecules, no energy is released. Energy is absorbed. So anabolism is when the molecules are synthesized and the energy is absorbed. Catabolism is when the molecules are broken down and the energy is released. Clear?
Yes.
During the process of respiration, oxygen is utilized and carbon dioxide, water, and energy are released as products. The combustion reaction requires oxygen. We know because combustion happens in the presence of O2 only. Some cells live where oxygen may or may not be available. Now, there are sufficient reasons to believe that the first cells on this planet lived in an atmosphere that lacked oxygen. Even among present day living organisms, some of that are adapted to anaerobic conditions. See, initially, when the earth was formed, oxygen was not there. That is why prokaryotic organisms are the first inhabitants of the earth, because they were mostly anaerobic, right? All the monerans, they were anaerobic in nature. So some of these organisms are facultative anaerobic, meaning they can respire, they respire aerobically. Aerobically means they respire in the presence of oxygen. But if just in case the oxygen is not available, then they can respire anaerobically, meaning without oxygen as well. So those organisms are known as facultative anaerobic. And those that are strictly, for example, there is an organism that will strictly and like only respire when oxygen is not present, then that becomes obligate anaerobe. Or if it respires when oxygen is present and strictly, then that becomes obligate aerobe. Okay, so obligate and facultative, two words that you should remember. Clear? Do you know the difference now?
Yeah.
Now, all the living organisms retain the enzymatic machinery to partially oxidize, meaning break down the glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is known as glycolysis. Now, see, glycolysis is that one breakdown, one kind of breakdown that is present in prokaryotes also and eukaryotes also. During this process, what happens is that glucose molecule is broken down partially, stepwise, and it happens in the absence of oxygen. Now we're going to see what glycolysis is about. Now the glyco is referring to the sugar and lysis means the splitting or the breakdown. The scheme of glycolysis was given by Gustav Emden, Otto Meyerhoff, and J. Parnas, and is often referred to as EMP pathway. So the other name for glycolysis is EMP pathway, and this EMP comes from their surnames, Emden, Meyerhoff, and Parnas. Now, in anaerobic organism, it is the only process in respiration for anaerobic, the ones that do not require oxygen. Glycolysis. Now, where does this happen? What is the site of glycolysis? So, glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. It occurs in the cytoplasm. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. So, at the end of glycolysis, two molecules of pyruvic acid are formed. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis, or from storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase. Meaning the plants, they take in, like how, for example, if we want to cook something, first we take out the ingredients from the refrigerator or wherever it is there, right? Same thing the plant is also doing. The plant is taking the stored glucose, which is there in its body, and then that glucose is actually derived from sucrose, because sucrose is how it is stored in the plant's body, sucrose or starch. and it is then converted into glucose and fructose, and any reaction that happens, it happens with the help of an enzyme, and the enzyme that is causing this conversion is invertase. Now, these two monosaccharide is because you cannot break down glucose into its monomeric unit or fructose into its monomeric unit. So that is why they are monosaccharides. Now, glucose or fructose, they would enter the glycolytic pathway. Now, glucose and fructose are phosphorylated. Phosphorylated means phosphate groups are added to it to give rise to glucose 6 phosphate by the activity of the enzyme hexokinase. So the glucose gets turned into glucose 6 phosphate because phosphate group is being added, and that happens in the presence of enzyme hexokinase. This phosphorylated form of glucose then isomerizes, changes its form to produce fructose 6 phosphate. Okay, so from glucose 6 phosphate, fructose 6 phosphate is formed. Subsequent steps of metabolism of glucose and fructose are same. All these steps that happens later on, it is same for both. Now, in glycolysis, a chain of 10 reactions under the control of different enzymes take place to produce pyruvate from glucose. See, pyruvate and pyruvic acid, both are the same thing. So don't get confused. Pyruvic acid and pyruvic, it's the same. Now, while studying the steps of glycolysis, please note the steps at which utilization or synthesis of ATP or NADPH, NADH2 takes place. Okay, so in each of the steps, we have to note where ATP is being synthesized or ATP is being consumed. For example, if we look at the step wherein glucose is getting converted into glucose 6 phosphate, what do you think? Is the ATP being synthesized or is it being produced in the first step?
Synthesized.
See, see it carefully. It is actually being released. I mean, ATP is getting broken down, it is being used, right? It is not getting synthesized because if you look at the arrow, from ATP it is going to ADP. And as I told you, when ATP is broken down, meaning when it is released, right? I mean, when it is actually utilized, not released, utilized. And in that way, the ATP gets converted into ADP. Okay, so here it is being synthesized. But if it will be shown like this, from ADP to ATP, when ATP has the arrow towards it, in that point of time, the ATP will be synthesized. Clear? Here,
Yes.
ATP will either be utilized for the process because, you know, some reactions can be the reactions that require energy. And energy again will come from ATP only. So somewhere in these reactions, ATP will be utilized. Somewhere it will be synthesized. Now, ATP is utilized at two steps. First, in the conversion of glucose into glucose 6 phosphate, and second, in the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. In this step and in this step, ATP is being utilized. Okay. Now, fructose 1,6 bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglycerol aldehyde. Meaning when we broke down or when we split the 1,6 bisphosphate, after fructose 6 phosphate has been formed, then it gets converted into fructose 1,6 bisphosphate, meaning 2 phosphates are there. Then this is actually broken down into 3 components because we know that fructose and glucose, both of them are 6 carbon compounds. So they can be broken down into 2 molecules of 3 carbon each, right? So what is that? It is glycerol aldehyde phosphate or dihydroxyacetone phosphate. And these two forms are interconvertible. They can be converted into each other. And both of them are 3 carbon compounds. Okay, we find that there is one step where NADH plus H plus is formed from NAD plus. This is when the 3-phosphoglycerol aldehyde is converted to 1,3-bis-phosphoglyceric acid. Now see, when this 3-carbon molecule is made, that is phosphoglycerol aldehyde, now that gets converted into bis-phosphoglyceric acid. During this point of time, the NADH2 is being synthesized because you see the arrow is towards the NADH2. Now, if you look at this condition, is the ATP being synthesized or is it being released? I mean, it is being utilized.
It's synthesized.
Yes, it is being synthesized. All right. Now, after the phosphoglycerol aldehyde has been made, it gets converted into bis-phosphoglyceric acid, and then that gets converted into phosphoglyceric acid, meaning one phosphate gets removed. That is why the one phosphate is going to this ADP and the ATP is being synthesized. Then the phosphoglyceric acid, that is PGA, gets converted into phosphoglycerate, and then the phosphoglycerate will be converted into phosphoenol pyruvate, and lastly it will be converted into pyruvic acid. Now the two redox equivalents are removed in the form of two hydrogen atoms from phosphoglycerol aldehyde and transferred to a molecule of NAD plus. That is why the NAD is getting converted into NADH. Phosphoglycerol aldehyde is oxidized with inorganic phosphate to get converted into bis-phosphoglyceric acid. The phosphoglyceric acid here is getting converted into bis-phosphoglyceric acid. The conversion of bis-phosphoglyceric acid to three molecules like 3-phosphoglyceric acid, PGA, is also an energy yielding process, meaning in this point of time, the ATP is being formed, and that means energy is being yielded. Now, another ATP is synthesized during the conversion of phosphoenol pyruvate into the last, that is pyruvic acid. So ultimately, if you look at here, so here one ATP is being synthesized and another ATP is being synthesized. So two molecules of ATP are synthesized. Here, one molecule of ATP is being utilized and another molecule of ATP is being utilized. So two molecules are utilized, two molecules are synthesized. So what is the net gain of ATP?
Zero.
Zero, correct, because the amount that is being synthesized is the amount that is being utilized. Now, pyruvic acid is then the key product of photosynthesis. What is the metabolic fate of pyruvic? Meaning, okay, we understand that okay, glycolysis happened in the cytoplasm. Now, what will happen to this pyruvic acid and what is its importance? So, actually, the pyruvic acid goes into several different directions depending upon what the cell would require. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. What are the three steps? First, we have lactic acid fermentation. Second, alcoholic fermentation. And lastly, aerobic respiration. So if we, you can say, classify into a very general term, so this pyruvic acid will actually undergo two processes. It can either go aerobic respiration or anaerobic. Now, under the anaerobic, there are two. Anaerobic means in the presence of oxygen or in the absence of oxygen.
Absence.
Absence of oxygen. So the anaerobic processes that the pyruvic acid undergoes are either lactic acid fermentation or alcoholic fermentation. Now, fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes in their body. For the complete oxidation of glucose to CO2 and H2O. However, organisms adopt Krebs cycle, which is also called as aerobic respiration, which requires O2 supply. Now, for organisms that are higher organisms, they will have this thing, aerobic respiration, also known as Krebs cycle in their body. But the lower organisms, they will have this particular thing, which is known as fermentation. It can happen in prokaryotic organism as well as unicellular eukaryotic organism. Clear?
Yes.
Can you give me an example of a unicellular eukaryote? An example of unicellular eukaryote. Hmm? Have you heard about yeast?
Yes, ma'am. It's unicellular fungi.
Correct. And fungus, all the fungi, they are eukaryotic. But they are multicellular eukaryotic, but the only exception is the unicellular organism, that is yeast Saccharomyces cerevisiae. Now, talking about fermentation. Up until now, glycolysis is clear.
Yes, ma'am.
Okay. Now,
Ma'am, what sort of question would be asked from this part? Like, should we learn the green table thoroughly?
from glycolysis, you first of all you learn the whole reaction only, because it's
Hmm.
very important. This is the only step in the book that has been defined properly, like all the steps. And so you read, learn this also, and you should know at which point ATP is being utilized, at which point ATP is being synthesized. That's important. And at what point NADH2 is being synthesized or utilized, whatever. Okay, because they can ask you, like, during the conversion of this, what happens? They'll give you 4 statements, they'll ask you to mark out the right one. Okay.
Yes, ma'am.
Now, moving on to fermentation. Now, in fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by sets of reactions where pyruvic acid is converted into CO2 and ethanol. The enzymes that are pyruvic acid decarboxylase, very important, the name of these two enzymes, and alcohol dehydrogenase. They are helping in the process of fermentation, because as I told you, no reaction will take place without the help of enzymes. So for this fermentation process, which is an anaerobic process, which at the end of which CO2 and ethanol are synthesized. So the pyruvic acid decarboxylase and alcohol dehydrogenase, these are the two enzymes for the catalyst. Other organisms, like some bacteria, produce lactic acid from pyruvic acid. The steps involved are shown in animal cells also, like muscles during exercise. When oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid. See, in our muscle cells, whenever we do some heavy exercises or we run or anything like that, like workouts and stuff, we feel like we are running out of air, because, at that point of time, the anaerobic condition arises for us, but because oxygen shortage happens. So in that time, in our muscles, lactic acid fermentation happens, wherein the pyruvic acid gets converted into lactic acid in our muscles. Now, the reducing agent, which is agent is NADH2, which is oxidized to NAD plus in both the processes. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of energy, again important. How much energy is released in these two processes? So less than 7% of the energy is released, and not all of it is trapped as to form the ATP. Also, these processes are very hazardous because either acid or alcohol is being formed. So both of these processes are dangerous. And see this thing. The glucose, after getting converted into pyruvic acid, can either undergo lactic acid fermentation, because of which lactic acid will be formed. Or it can go into ethanol formation, meaning alcoholic fermentation. So either alcohol or lactic acid will be formed. And in both of these steps, if you see, the NADH2 is actually being utilized to form NAD+. So energy is again being used only. It's not being released. Not much energy is released in this step. Now what happens is that the yeast, after they are done doing the process of fermentation, they will poison themselves, like they will die, because if the concentration of alcohol reaches to 13%, then the yeast cannot survive and they die. So again, a very unique, you can say, character comes into mind is that, for all those alcoholic beverages that are sold in the market, for their fermentation, because alcoholic beverages are made by fermentation only, fermentation of rice water, fermentation of potato and whatnot. So in that fermentation process, yeast is used only, because baker's yeast, that is also a product of fermentation. Then all the drinks, the alcoholic drinks, they are produced, yeast is. Okay, now this yeast will actually poison itself when the concentration of alcohol in any drink or any food substance reaches to 13%. So naturally, we can understand this one thing, that in all the alcoholic drinks that are naturally fermented, meaning that are made after natural fermentation, in them, what will happen? The maximum percentage, or you can say the maximum concentration of alcohol will be 13%, not more than that. Clear? Any doubts
Yes,
about
ma'am.
me?
No.
Now they have given a question also. How do you think alcoholic beverages of alcohol content greater than this 13% concentration are obtained? So the answer is distillation. Now this distillation process happens in, like, wherever wineries and places where wines and alcohols are synthesized. So distillation is the process by which a greater concentration of alcohol will be there. But if someone is making alcohol naturally, then only 13%. Now, what then is the process by which organisms can carry out complete oxidation of glucose and extract the energy to stored to synthesize a large number of ATP molecules? Now, for us higher organisms, for all the eukaryotes, like the multicellular eukaryotic organisms, they need oxygen. We know that. And they need oxygen for respiration or release of energy. And that process is aerobic respiration. It is the process that leads to the complete oxidation. Now, up until now, we have studied about partial oxidation or partial breakdown. But right now, we are talking about complete oxidation of any kind of organic substance, and that happens in the presence of oxygen, and it releases CO2, water, and a large amount of energy present in the substrate. So aerobic respiration is the most common in multicellular eukaryotic organisms. Moving forward, there is aerobic respiration. Now for aerobic respiration to take place within the mitochondria, the final product of glycolysis. Now, where is the glycolysis? It is there in the, it is happening in the cytoplasm. So, if the glycolysis, like at the end of glycolysis, if pyruvic acid is formed, that will also be there in the cytoplasm only. So, from the cytoplasm, the pyruvic acid gets transported into the mitochondria, because the site of aerobic respiration is mitochondria. Clear? The first thing that will happen is that the pyruvic acid will get actually transported from the cytoplasm into the mitochondria. Up until now, do you have any doubts? Sure.
Yes.
Okay. Now talking about the crucial events in aerobic respiration. The complete oxidation of pyruvate by stepwise removal of all the hydrogen atoms, leaving 3 molecules of CO2. So complete oxidation happens by stepwise removal of hydrogen atoms. Then the second step, the passing on of the electrons removed as a part of hydrogen atom to molecular oxygen with simultaneous synthesis of ATP. So electrons are also passed. Now, the first process wherein the hydrogen atoms are being utilized, or you can say removed, this part takes place in the matrix of the mitochondria. Because if you remember, we studied in the cell chapter, the mitochondria has a double membrane. One is the outer, one is the inner. Now, the inner one encloses the matrix. And the second process, it happens is like it is located on the inner membrane of the mitochondria. So both of these processes, which are part of aerobic respiration only, and yes, 100% it will happen in the mitochondria, but there will be different sites. Any doubts up until now?
No.
Okay. Now, pyruvate or pyruvic acid, which is formed by the glycolytic catabolism of carbohydrates in the cytosol. See, cytosol is another word for cytoplasm. After it enters the mitochondrial matrix, undergoes oxidative decarboxylation. Now, this is a step that happens, meaning the pyruvic acid, as soon as it enters the mitochondrial matrix, it undergoes a process which is known as oxidative decarboxylation. Now, this again happens with the help of certain enzymes, and that enzyme is actually pyruvic dehydrogenase, meaning here the pyruvic acid gets converted into acetyl coenzyme A with the help of the enzyme that is pyruvic dehydrogenase, and it happens in the presence of magnesium dipositive ion. So the reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes, including NAD plus and coenzyme A. Why? Because to aid the process or to make the process happen flawlessly, it requires certain, you can say, cofactors. Clear? Hmm?
Yes.
During this process, two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid. Acetyl coenzyme A, the two molecules of pyruvic acid gets converted or they are produced from one molecule of glucose. Now, the acetyl coenzyme A then enters a cyclic pathway, which is known as tricarboxylic acid cycle, TCA, and it is also known as Krebs cycle, after the scientist Hans Krebs. He was the person who actually found this out and told us about it. Now talking about the tricarboxylic acid cycle, or this cycle is also known as citric acid cycle, because the first molecule that is formed is citric acid. So that is one. So it has three names: tricarboxylic acid cycle, citric acid cycle, or the Krebs cycle. Now, the tricarboxylic acid cycle starts with the condensation of acetyl group with oxaloacetic acid and water to yield citric acid. The first molecule that is produced is citric acid. If you look at this diagram, this oxaloacetic acid combines with the acetyl coenzyme A and water to form citric acid. Clear? This is the first step. The reaction is catalyzed by the enzyme citrate synthase, and the molecule of coenzyme A is released during this step. Citrate, or the citric acid that is formed, it gets converted into isocitrate. Okay, this citric acid is formed, it gets converted into isocitrate, and later on it gets converted into alpha ketoglutaric acid. See, these are the sub-molecules that are formed during the whole entire cycle. They have to be formed. Now, it is followed by two successive steps of decarboxylation, leading to the formation of alpha glutaric acid. And then from alpha glutaric acid, it forms the succinic acid. From succinic acid, it will form succinyl coenzyme A. Now, in the remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to oxaloacetic acid. Again, it forms the oxaloacetic acid because ultimately it is a cyclic process. And if oxaloacetic acid cannot be synthesized, then the cycle will not keep on going. So that the cycle keeps on going, the oxaloacetic acid is synthesized again. Now, during the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. GTP is just like ATP only, but just like how ATP has adenosine, GTP has guanosine. And this process wherein the GTP is being synthesized from the conversion of succinic acid, from the conversion of succinyl coenzyme A to succinic acid, we'll call it as substrate level phosphorylation, because ultimately they are the substrates during the reaction. And phosphorylation means formation of ATP. We have studied this in photosynthesis. Now, in a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus, and one where FADH2 is formed from FAD plus. The continued oxidation of acetyl coenzyme A via the TCA cycle requires the continued replenishment of oxaloacetic acid again, so that the cycle keeps on going uninterrupted. So that is why from the succinyl coenzyme A, then succinic acid, and then many other acids are formed to form lately the oxaloacetic acid. In addition, it also requires regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows. Now, this is the whole entire summary for the reaction of Krebs cycle. That is, pyruvic acid along with 4 NAD plus, 1 FAD plus, 2 molecules of H2O and ADP plus inorganic phosphate. These are the reactants that are required. It happens in the mitochondrial matrix, and ultimately 3 molecules of CO2, 4 NADH, 4 H plus, FADH2 and ATP are synthesized. So this is the TCA or the citric acid cycle or the Krebs cycle. Do you have any doubts up until now?
No.
Sure?
Yes.
Okay. Now, we have till now seen that glucose has been broken down to release CO2 and 8 molecules of NADH plus H plus, 2 molecules of FADH2 have been synthesized besides just 2 molecules of ATP in TCA cycle. Always remember that 1 molecule of NADH2 can give rise to 3 molecules of ATP, and 1 molecule of FADH2 can give rise to 2 molecules of ATP. This is how it happens. Now, you may be wondering why we are discussing it at all. See, all of these are chemical intermediates. They are very important for ATP synthesis, and we will talk about it when we talk about electron transport system and oxidative phosphorylation. Always remember, whenever anyone says phosphorylation, it means synthesis of ATP. Now, the following steps in the respiratory processes are to release and utilize the energy which is stored in the NADH2 and FADH2. This is accomplished when they are oxidized through the ETS, that is electron transport system, and then their electrons are passed on to the oxygen, resulting in the formation of water. That is how the electron transport happens in the, you can say, the cell machinery. Now, the metabolic pathway through which the electron passes from one carrier to another is known as electron transport system. Now, where it happens? So, it is present in the inner mitochondrial membrane. Now, electrons from the NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by an NADH dehydrogenase. This is the complex one. So, from the NADH, okay, or you can say the NADH dehydrogenase, it receives the electrons from the citric acid cycle. And from that, the electrons are then transferred to the ubiquinol. It is again a molecule which is located within the inner membrane of the mitochondria. Apart from this electron which the ubiquinol has received, it also receives the electron from the FADH2, which is the second complex. And once it receives the electrons from both the sides, it gets reduced. Now it gets reduced and it is now ubiquinol. Then it is oxidized with the transfer of electrons to cytochrome C via cytochrome BC1. So it transfers its electron to cytochrome C with the help of cytochrome BC1. See this thing. Cytochrome C is here, right? And cytochrome C is here. Now this cytochrome C is receiving the electrons from the ubiquinol from, or you can say via cytochrome D. Cytochrome C is a small protein attached to the outer surface of the inner membrane and it acts like a mobile carrier. Mobile means it can move from one complex to another complex, and because of this property of being mobile in nature, it can transfer the electrons between the complex 3 and the complex 4. It is movable. It goes there and there. It happens sort of like a moving mechanism. Now, the complex 4 refers to cytochrome C oxidase complex containing the cytochrome A and cytochrome C, the A3. You see, the cytochrome C, it has two complexes. It has A and it has A3, which makes it like one big complex. And then apart from A and A3, it also has the two copper center. Again, for the aiding for the electron transport only, they have the copper centers present. Up until now, do you have any doubts in the ETS?
No.
Now, when the electrons pass from one carrier to another via complex 1 to 4 in the ETS or the electron transport chain, they are coupled to ATP synthase. Now, see, ATP synthase is the name of the complex 5. And this complex is responsible for the formation of ATP from the ADP and inorganic phosphate. Now, the number of ATP molecules synthesized depends on the nature of the electron donor. If the electron donor is giving more and more electrons, then that ultimately means that more and more ATP will be synthesized. For example, if you are talking about NADH, so one molecule of NADH, when it gets oxidized, it will give rise to 3 molecules of ATP. But if we oxidize one molecule of FADH2, then that will produce 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. See, all of these steps, okay, 100% it has to happen in the presence of oxygen, but there is no role of oxygen over here. It's of no use. But it happens to be of use at the last stage, at the end. So oxygen presence is very important since it drives the whole process of removing hydrogen from the system. Because all of these, you can say removals that we are like witnessing right now, they are happening because oxygen is present. Now, final hydrogen acceptor is the oxygen. So oxygen acts like the final hydrogen acceptor. Now, unlike photophosphorylation means the synthesis of ATP which happens in the presence of light. So unlike that, where it is the light energy that is utilized for the production of proton gradients required for photophosphorylation. You remember chemiosmotic hypothesis?
Yes.
So after chemiosmotic hypothesis, we know that how the substrate level or the photophosphorylation happens. But unlike that, in the respiration, it is the energy of oxidation and reduction, means redox, that is used for the formation of ATP. That is why it is known as oxidative phosphorylation. Meaning, because of the oxidation processes, the ATP is being synthesized. Now, you have already studied about the mechanism of membrane-linked ATP synthesis in chemiosmotic hypothesis. Now, the energy released during the electron transport system is utilized in the synthesizing ATP with the help of ATP synthase, that is complex 5. The name of the complex 5 is ATP synthase. This complex consists of two major components. It has F1 and F0. We know that whenever the electrons or the, you can say the protons from the lumen, right, wherever the gradient of proton is more, whenever it passes from the F0 to F1, it is actually coupled to the synthesis of ATP. Now, the F1 is known as the head piece. It is a peripheral membrane protein complex. We know that proteins are of two types, peripheral and integral. So, the F1 is the peripheral membrane protein, and it contains the site for synthesis of ATP from ADP and inorganic phosphate. The F0, it is an integral protein. It is present like embedded below. So, it forms a channel through which the protons can pass. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, two protons or two H plus ions pass through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. Down the gradient means at a place where hydrogen ions or the protons were present in much more amount, and then there is one place where there is not enough. So this creates a gradient, and the passing of this gradient happens, and then the ATP is synthesized. Do you have any doubts up until now?
No.
ETS, chemiosmotic hypothesis, glycolysis, fermentation, all of these processes are clear to you?
Yes.
Sure?
Yeah.
Okay. Now we have the respiratory balance sheet. Now what we do when we make in general a balance sheet, we put out how many numbers are, like what are the outputs, we put the inputs and everything. So same way, when we have to calculate, here money is not the issue. Here the money is the ATP, right? Because it is the energy currency. So in order to find out how much ATP is synthesized, how much of it is released, we make a respiratory balance sheet. So for every glucose molecule extract, like oxidized, right? For like whenever the glucose molecule gets oxidized, then ATP is being formed. Now there is a sequential orderly pathway functioning with one substrate forming the next, and with glycolysis, PCA cycle, ETS, one after the other, all the pathways are happening. The NADH synthesized in glycolysis is transferred into mitochondria and undergoes oxidative phosphorylation by aerobic respiration. None of the intermediates in the pathways are utilized to synthesize any other compound. It is just primarily concerned with only and only ATP synthesis. Only glucose is being respired, no other alternative substrate. Like these are the, you can say, assumptions we are creating of a very ideal condition, which is not possible in the living organism, because there are so many steps that happens in the body, and nothing is so perfect. Like all of the things are happening at once, so we cannot consider like the ideal condition. So this kind of assumptions are not really valid in a living system. All pathways work simultaneously and do not take place one after another. Substrate enter the pathways and are withdrawn from it, and whenever it is necessary, then only the substrates are withdrawn and made to synthesize ATP. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. Yet, it is useful to do this exercise to appreciate the beauty and efficiency of the living system. So there can be a net gain of 36 ATP molecules during aerobic respiration of 1 molecule of glucose. Very important to remember. How many molecules will be synthesized? So 36 molecules will be synthesized. Now let us compare the fermentation alongside aerobic respiration. Fermentation accounts only for partial breakdown of glucose, whereas in aerobic respiration, it is completely degraded to CO2 and H2O. This is the major difference. This is important. The major difference between this thing and the fermentation and aerobic respiration is that the first process happens in an anaerobic condition, meaning in the absence of oxygen. The second, obviously, the name suggests it's happened in the presence of oxygen. After that, in the fermentation being anaerobic, here the partial breakdown of glucose happens, whereas for aerobic, the complete breakdown happens. Now, in fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid. Net gain is 2 molecules, whereas many more molecules of ATP are generated under aerobic conditions. NADH is oxidized to NAD plus rather slowly in fermentation. Slowly it happens. However, it is very vigorous in the case of aerobic respiration. So these are the very important differences between the fermentation and aerobic respiration. Now we have amphibolic pathway. See, there is catabolic, there is anabolic. But when both of the things are happening, meaning energy is being utilized also in any process, molecules are synthesized. So that is anabolic. And then energy is also released and molecules are broken down. So for respiration, it is amphibolic because it is a sum total of catabolic as well as anabolic. Now, glucose is the favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrates can also be respired, as has been mentioned earlier, but they do not enter the respiratory pathway at the first step. All see, no matter what the molecule is, whether it is fat, whether it is other than the glucose, it has to be converted into glucose, then only it can enter the whole respiration. Now, the fat molecules would need to be broken down into glycerol and fatty acid. If fatty acids were to be respired, they would first be degraded to acetyl coenzyme A and then enter the pathway. So fat, when it will be broken down into fatty acid and glycerol, the fatty acid part will get converted into acetyl coenzyme A and then the respiratory pathway will happen. The the glycerol would enter the pathway after being converted into phosphoglycerol aldehyde. The proteins would be degraded by proteases and the individual amino acids for deamination, depending on their structure, would enter the pathway at some stage within the Krebs cycle. So ultimately, we are seeing that no matter what substrate we are choosing, whether it is fat or protein, it has to be converted into glucose. Then only, or not just glucose and its form. Then only it can undergo the process of respiration. Now, since respiration involves the breakdown of substrate, the respiratory process has traditionally been considered as a catabolic process and the respiratory pathway as a catabolic pathway because breakdown of substrates are happening, release of energy is happening. But is this understanding correct? Because we have seen there are many molecules that are synthesized as well, right? So we cannot call it purely catabolic. So ultimately we'll call it amphibolic, but hence fatty acids would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as a substrate. But when the organisms need to synthesize fatty acid, this acetyl coenzyme A would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into picture both during the breakdown also and during the synthesis also, or more particularly right now we're discussing about the fatty acid. Similarly, during breakdown and synthesis of protein 2, respiratory intermediates form the link. Breaking down processes within the living organism is catabolism and synthesis is anabolism. Because respiratory pathways involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway. Amphi means dual, right? Amphibians, you remember, amphi means dual. So it is a pathway that has dual, you can say, catabolism and anabolism, like dual pathways are present. That is why we call it as amphibolic. We cannot just call it as catabolic. So this is how, for example, fats are there. They have to be converted into fatty acid and glycerol. And then this fatty acid will be converted into acetyl coenzyme A, and then only it will undergo the aerobic respiration. Same goes for the simple sugars such as glucose. Now we know that the glucose also gets converted into acetyl coenzyme A, but the glucose is the most favored substrate. Also, if proteins we are talking about, so when we break down proteins, we get many amino acids because one protein is made up of 20 amino acids. So after this amino acid has been synthesized, it will get converted into acetyl coenzyme A and ultimately reach the Krebs cycle. So again, the thing is to note that all the substrates, they can enter the respiratory pathway 100%, but not directly like they are. They have to be converted into glucose or they have to be converted into the acetyl coenzyme A, and then only it can enter the Krebs cycle or aerobic respiration. Any doubts up until now?
No.
Okay, now coming on to the, I think this is the last topic, that is respiratory quotient. Now, respiratory quotient. As you know, during aerobic respiration, O2 is consumed and CO2 is released. The ratio of the volume of CO2 evolved to the volume of O2 consumed in respiration is known as the respiratory quotient or the respiratory ratio, RQ, which is volume of CO2 evolved, the one that is being evolved and the one that is being consumed. The respiratory quotient depends upon the type of respiratory substrate which is used during respiration. When carbohydrates are used as substrate and are completely oxidized, because the carbohydrates are broken down to the whole molecule gets broken down. So in that case, the RQ value will be 1 because the amount of CO2 which is evolved will be equal to the amount of O2 which is consumed. Just like how here, if 6 molecules of O2 is taken in, then 6 molecules of CO2 is let out. So the RQ value for when carbohydrate is used is 1. But when fats are used in respiration, the RQ value will always be less than 1. For example, for a fatty acid that is known as tripalmitin, if that is used, then see, 145 O2 is used, but 102 CO2 is used. So up until it is less than the 1, right? So for like fat, always it will be less than 1. When proteins are used as respiratory substrate, the ratio would be about 0.9. These respiratory questions are important to remember, and in exam they can either give a different kind of name of a fat, and they'll give you for example, you don't know the name of the fat that is given in the question, and they did give you the respiratory value or the respiratory quotient. For example, they give you 0.2. So you'll know that it is, if it is less than 1, it is a fat that is being used as a substrate. Getting my point? And
Yes.
whenever proteins will be used, it will come out to be exactly 0.9. So this is about the respiratory quotient. And What is important to recognize is that in living organisms, respiratory substrates are more often, like more than one. Pure proteins or fats are never used as respiratory substrates. Pure ones are never used because it will be difficult to break them down. That is why the conjugated ones, the ones that have something involved in them, they will be used as a respiratory substrate. So this is like we have completed the chapter that is respiration in plants. Now you tell me, do you have any doubts in this chapter? This chapter is sort of lengthy also, and it has a lot of technicalities and calculation. So it is very, like strongly advisable for you to actually read the NCERT very nice. Now tell me if you have any doubt.
Let me just cross check.
Yeah, just check it once. Yes.
No doubt.
No doubts? Okay. Very good. All right. So we are done with this chapter and okay. You tell me, how is your preparation for NEET going?
Yes, ma'am, it's going well.
Going well?
Mostly, right now I'm just trying to focus on biology because I feel there's no time for physics.
Hmm. And how is this thing?
Chemistry.
Chemistry?
Yeah, that's going well.
Good. Okay. Like, are you able to solve all the questions and everything?
Yes, I mean that was completely related with both, so I'm able to do that.
Oh, very good. And what about biology? Are you able to solve questions in biology?
Yes, ma'am, I'm able to do it.
Okay, very good. All right. So after now this since this chapter is done, now we'll be starting with a new chapter and it is a very interesting chapter. Can you guess?
Yes, we're breathing.
Breathing we have already done. The chapter that I'm going to start, it is the body fluid and circulation. The most beautiful chapter and it is one of those chapters that is loved by many students. Do you like this chapter?
Yes, ma'am, it's probably my favorite.
Yeah, it is like literally loved by so many students. Even me as a teacher, I love to teach this because it is interesting. The students can relate to this chapter also, right? So now we don't have a lot of, you can say chapters left, but we do have, like, see, I am not worried about your 12th because I know that you have actually done it nicely, isn't it? It's just the class 11
Yes,
that is the problematic one, right?
yes, it's just 11.
Hmm. All right. Okay, so we are going to start with body fluids and circulation. Are you tired?
No, ma'am.
Now, body fluids and circulation. Now, first, the biggest thing that we are to know in this particular chapter is that every circulatory mechanism that happens in our body, it happens because of the presence of blood. And our blood is very famously known as the fluid connective tissue because it is obviously 100% liquid, right? And it is the connective tissue. Now, why is it the connective tissue? Because it is connecting all the different parts of our body. Now, you have learned that all the living cells have to be provided with nutrients, oxygen, and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have efficient mechanisms for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for this transportation. Simple organisms such as sponges, coelentrates circulate water from their surroundings through their body cavities to facilitate the cells to exchange their substances. More complex organisms use special fluids within their bodies to transport such materials. Blood is one such fluid. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid that is lymph. It is a colorless fluid that is used for the transportation of certain substances. In this chapter, you will learn about the composition and properties of the blood and lymph, which is also known as tissue fluid, and the mechanism of circulation of blood is also explained. Talking about the blood, it is a special connective tissue containing of, now what does the blood contains? It contains the fluid matrix, it contains the plasma, and it contains the formed elements, sort of like the cells that are present. Now talking about the plasma. Now plasma is a straw colored, yellowish, light yellowish colored, viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and protein, meaning plasma itself is made up of water and protein. It contributes to 6 to 8% of the plasma. Fibrinogen, globulins, and albumins are the names of the major proteins that are found in the plasma. Fibrinogens are used or needed for clotting or coagulation of blood. Very important. Fibrinogen. Then globulins. Globulins are involved in the defense mechanism because antibody, all of these are globulins. So these are involved in the defense mechanism. And then there is the albumins, which help in the osmotic balance, meaning the regulation of the electrolytes in our body that happens with the help of albumins. Plasma also contains small amounts of minerals such as sodium, calcium, magnesium, bicarbonate ion, chlorine, etc. All of these are also present in the plasma. Glucose, amino acid, lipids are also present in the plasma as they are always in transit in the body. In transit means they're always moving in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. When we remove the clotting factor from the plasma, we'll call the plasma as serum. So if you remove all the clotting factors from the plasma, now the plasma won't be called as plasma, it will be called as serum. Now talking about the formed elements. Erythrocytes, leukocytes, platelets are collectively called as the formed elements and they constitute nearly 45% of the blood. Erythrocytes are the red blood cells and they are the most abundant of all the cells in the blood. A healthy human has on an average 5 millions to 5.5 millions of RBC per millimeter cube of the blood. RBCs are formed in the red bone marrow in adults. Where does the formation of RBC takes place? So in the red bone marrow in the case of adults. RBCs are devoid of nucleus. They are enucleated in most of the mammals and they are biconcave in shape. They have a red colored iron containing complex protein which is known as hemoglobin. Hence the color and the name of the cells. Because they contain the red color pigment protein, hemoglobin, they are called as the red blood cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of the blood. These molecules play a significant role in the transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen. And the spleen is known as the graveyard of RBC. So the lifespan of RBC is just 120 days. After that, they are destroyed and they die. And then new RBCs are formed by the bone marrow, the red bone marrow. Now talking about the leukocytes. Leukocytes, they are the white blood cells. They do not have any pigment, that is hemoglobin is absent in them. They have nucleus. They are nucleated and they are relatively lesser in number, which averages to 6,000 to 8,000 per millimeter cube of blood. Leukocytes are generally short-lived. We have two main categories of WBC, granulocytes and agranulocytes. See, the WBC or the leukocytes can be divided into two types. The first one is granulocyte, the second is agran. Neutrophils, eosinophils, basophils. These are the different types of granulocytes, while lymphocytes and monocytes are agranulocytes. Neutrophils are the most abundant cells, that is 60 to 65% of the total WBCs. And basophils are the least, meaning 0.5 to 1%. This percentage is important to remember. Neutrophils and monocytes, that is 6 to 8%, are phagocytotic cells, or phagocytic cells, which destroy the foreign organisms entering the body. Basophils secrete histamine, serotonin, and heparin, and they are involved in the inflammatory reactions. Eosinophils, that is 2 to 3%, they resist infections and are also associated with allergic reaction. Lymphocytes, that contains about 20 to 25%. They are of two major types, B lymphocytes and T lymphocytes. Both B and T lymphocytes are responsible for immune responses of the body. Platelets are known as thrombocytes. These are the cell fragments produced from megakaryocytes, special cells in the bone marrow. So the platelets which our body has, it is known as thrombocytes, and it is cell fragments. It's not a whole cell, rather fragments of the cell. Now, blood normally contains 1,50,000 and up till 3,50,000 platelets per millimeter cube of the blood. Platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. So whenever we get injured, whenever we are hurt, we actually stop bleeding because of the presence of platelets, because platelets causes blood clotting, and then we stop bleeding, right? So all of these are the components of the blood. Now tell me if you have any doubts.
No.
Okay, do you know the name of the disease in which the number of platelets get reduced? Like, very seriously, the number of platelets gets reduced. Any idea? It is dengue. You know, whenever anyone has dengue fever, he or she will have the platelets count reduced to a very, very less number if it is very severe. Now, moving forward, we'll talk about the blood groups. As you know, blood of human beings differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. The two types of blood groupings that we based our study upon is the ABO blood grouping and the RH, and they are widely used all over the world. ABO blood grouping. It is based on the presence or absence of two surface antigens. Antigens are the ones that give rise to antibodies. Always remember that. And whether they are present on the RBC or not, we can find out the blood group. So, ABO blood grouping is based on the presence or absence of two surface antigens on the RBCs, namely antigen A and antigen B. Similarly, the plasma of different individuals contain two natural antibodies, proteins produced in response to antigen. The distribution of antigen and antibody, they are given here. For example, if it is blood group A. So blood group A will have antigen A, but will have anti-B antibody. Blood group B will have antigen B, but it will have anti-A antibody. Then there is blood group AB. It has the antigen for both, antigen A, antigen B, but it will not have any antibody, meaning no antibodies will be produced against any of these because both the antigens are present on one RBC. Now talking about the O blood group, they will have no antigen. If no antigen is produced, then they will have the antibody for both anti-A and anti-B. So for all these blood groups, for example, if we are talking about blood group A, now the blood group A can take the blood from the A blood group or the person who has O blood group. Why? Because the O blood group does not have antigen at all. Now the B one, they can take blood from the B blood group people or the O. So because of this very reason, the O is known as the universal donor. It can donate blood to anyone because it does not have any antigen present on it. And if antigens are not present, then antibodies will not be made. Talking about the blood group AB. Now the AB will have, or it can, you can say, get the blood donated from the blood group AB, A, B, and O. So in terms of taking the blood or donating the blood, the AB blood group is known as the universal recipient because it can take blood from any blood group. And the O, it is known as the universal donor because it can donate to any blood. Now, the second type of blood grouping system is the RH grouping. Another antigen, that is the RH antigen, similar to the one present in the rhesus monkeys, hence the name RH is coming. It is also observed on the surface of RBCs of majority, nearly 80% of humans. Those that have the RH antigen present, they are the RH positive individuals. And those that do not, they are the RH negative individuals. Now, an negative, if exposed to RH positive blood, will form specific antibodies against the RH antigens. Because, it is against like plus and minus situations, they're cancelling out each other. Therefore, RH group should be matched before the transfusion. It doesn't mean that, okay, a person with A blood group and then there is a person with another blood group. Now they can donate blood to each other. No, they have that their RH factor or RH grouping have to be matched because maybe one person is A positive, the other person is A negative. So now no blood transfusion can happen. A special case of RH incompatibility, that is mismatching, has been observed between the RH negative blood of a pregnant mother. For example, a female who is pregnant, she is A negative, RH negative, and the baby she is carrying, the fetus that she is carrying, it is RH positive, meaning it is maybe B positive or A positive. Now, the RH antigens of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy because we know that the bloods of the mother and the fetus, they are separated by the placenta. However, at the time of delivery, the first child, there is a possibility of exposure of the maternal blood to the small amount of RH positive blood from the fetus. Maybe due to some reason, the mother's RH negative blood can get exposed to the RH positive of the fetus. Now, in the mother's body, quickly antibodies will start being formed. Now, antibodies are formed. Now, if in the next pregnancy, the RH antibodies from the mother, which is who was RH negative, this could be fatal because, if the next baby is also RH positive, okay, which is against the mother's RH negative. So the antibodies can actually start attacking the fetus RBC, and this could be fatal. It could lead to the death of the baby, or it could cause severe anemia because RBCs of the baby will be destroyed. And also jaundice can also be there in the baby. So this condition is known as erythroblastosis fetalis. Erythrocytes, they're getting destroyed in the fetus situation. So what can we do to avoid this situation? No one can control whether the child is going to be positive blood group or the negative. No one can control that. So what can be done? If, for example, in the first pregnancy, if the mother is RH negative and the child is positive, then as the birth is done, so this can be avoided by administering or giving anti-RH antibodies to the mother immediately after the delivery of the first child. Meaning RH antibodies will be formed in the mother if she is exposed to the blood of the RH positive baby. If we give anti-RH antibody, anti to this, then this will be cancelled out. Now in the next pregnancy, even if the baby is RH negative, then also nothing will happen. I mean, if the baby is RH positive, then also nothing will happen. Did you understand what is this RH grouping and incompatibility?
Yeah. Yes, ma'am.
And why is it important to actually match the blood group so precisely so that nothing of this can be happening? Clear?
Yes, ma'am.
Okay. Now let's just keep it till here only for today. In the next class, we'll start with coagulation of blood. Up until now, do you have any doubts?
Yes, ma'am. Can you take the blood? Yes, ma'am. This part. Ma'am, you said that B blood group can get blood donated from B only, right? B and O.
Hmm.
But O also has antibody A, which can kill the uh, no. O also has antibody B, which can kill the B antigen, right?
Yeah, but see, since the O does not have any antigen, so it doesn't matter what antibody it carries. Antibodies are always synthesized against a particular antigen. If, for example, B has antigen B present on it, so that antigen will synthesize the antibody for the blood group that is against it, that is anti-A. But the O does not have any antigen at all, meaning it does not have the property to synthesize any antibody against anyone. It cannot synthesize on its own. Why? Because antigens are absent. Always remember that antigens will give rise to antibodies. Whenever any antigen is present, then only the antibody can be synthesized. This is the reason why the O blood group can donate to every single other person, but it cannot take from any. Clear?
Yes, ma'am.
And see, they have written here anti-A and anti-B. This is for its own thing. That it cannot take A or B blood because it has the antibodies already present in for its support or for its protection. But if it is donating, then the antigens are active, right? So that is why it can donate, but it cannot receive other than the O. Clear?
Yes, and one more question, ma'am, on this. But you said AB can receive from all of them, right?
Hmm.
So let's say AB receives from B, then B has antibody A in them.
Hmm.
So won't that change the constitution of blood in AB?
No. See, A and B, both of them are different, and AB is a combination of them both because it is codominance. This is not A separately, B separately. It is AB together. So
Yes.
anti-A it has. Okay, if
Hmm.
B is given, like B is donating blood to AB, it has anti-A. It will not affect AB because it is AB, not A. Had it been just A, then it will. Getting my
Okay.
point?
Yes, got
Yeah.
it.
Nor will it affect the B separately. It has to affect it together because it is the expression is together. Clear?
Yes, ma'am. Clear.